infectious diseases and molecular genetics and microbiology, and a staff physician at the Durham VA. He got his MD, PhD at Columbia, and then went to Duke University for residency and fellowship training. And as a Carolina grad, I will just give one go Tar Heels to that, and I will not fight with you over it for the rest of the evening. But you know how I feel about Duke. He then joined the faculty as an assistant professor at Duke University, where much of his research focused around biomarkers and transcriptional profiling of infectious diseases, specifically looking for markers that could differentiate bacterial, viral, and non-infectious diseases in a rapid manner. And now at Danaher, he focuses on host responses to infections. This work largely employs transcriptomics, but also epigenomics, metabolomics, and proteomics. Areas of clinical interests are similar to the past, and they include the ability to diagnose infection, to differentiate bacterial from viral infection, and to understand the heterogeneity of sepsis to enable precision medicine approaches. So with no further ado, please help me welcome Dr. Salik, and I'm really excited for your talk. Thanks for joining us.
Thank you very much, Kyle. That was great, and thank you all for being here. You had said it was going to be recorded. Is that already happening? Okay, all right, great. Just want to make sure I didn't have anything else to do. When Kyle said that there was going to be food here for people in advance, I just assumed that half of you were here for the food and half of you might be here for me. But since I don't know who is here for what purpose, I'm just going to assume you're all here for me. So thank you all for coming out and braving the cold. So as Kyle mentioned, I'm currently at Danaher, and I've been here for almost 2 years at Danaher, and before that, you're sort of living in the academic life. I had the opportunity to talk with some of the students about kind of my journey to where I am, which I won't belabor at this point, but I think some of that will come through in the context of the presentation that I'm going to give you today. Just very briefly about Danaher. Start my video. There we go. Okay, so I've heard it described to me that Danaher is the largest company you've never heard of. $165 billion with about 70,000 employees. Huge company. But again, historically, Danaher was a name that generally tried to stay in the background, and they let their operating companies, what we call opcos, kind of speak for themselves. I'm assuming that most of you have probably heard of some of these operating companies. Danaher actually used to be an even larger company. They had a whole other division focused on like product identification and water purification. Pantone, the color company, was a Danaher company, oddly enough. And then a few years ago, Danaher made a strategic decision to really become a science and technology company. So they actually siphoned off all of those other companies to a completely separate publicly traded company, and everything that remains really fits into one of these 4 categories of biotechnology, life sciences, diagnostics, and genomic touch points across the board, but generally focused on diagnostics, where there are 6 diagnostics companies. And as part of this transition to a science and technology company, they realized that they needed some overarching vision of what direction the corporation, Danaher, should go in. Historically, all of that directionality and strategy was defined by the individual company. So if you were at Cepheid working on sepsis and you wanted to talk to somebody at Beckman working on sepsis, you needed a confidentiality agreement to have that conversation. And now those barriers are coming down. These companies aren't going away. The brands are still there, but we're having a lot more visibility and crosstalk. And that's part of my remit, is to facilitate that and to think about infectious diseases everywhere it exists for future direction. So that's a little bit about the role that I play within Danaher, and hopefully, again, you'll have a little bit more familiarity with Danaher than you did coming in today. So we're going to talk about infection. So what is an infection? That seems pretty obvious to all of us. We've all had an infection of one sort or another, but when it comes down to actually defining it, it becomes a little bit more challenging, and there's a lot more nuance and gray. But at the core of it, you need really 3 things to have an infection. You need a host to harbor the infection, and a susceptible host. There's certainly certain conditions that impart a higher susceptibility. The very young, the very old, the immunocompromised, but in truth, all of us are susceptible as hosts. You obviously need a pathogen to cause the infection. This is between the type of pathogen that might cause an infection and the type of host that might harbor the infection. But that's not enough to have an infection because we all harbor pathogens in and on our bodies, but we are not walking around in a constant state of infection. So what you need is you need clinical illness. You need some kind of symptom, some deviation from equilibrium that manifests in clinical disease. And so it's really the combination of those two elements, but the interaction between them that ultimately results in an infection. So that said, how do we know, clinically speaking, if an infection is present and ultimately how to treat it? And historically, certainly technology has evolved from the days of Koch and his Koch's hypothesis and the microscope and microbiology. I think he probably really invented clinical microbiology in at least the modern vision of it in terms of culture. And our technologies have enabled or expanded our ability to identify pathogens. And sequencing is really now stepping into the forefront in terms of what its capabilities are. We were talking before during the time before this about sort of the expectation that NGS is going to continue to come down in cost, continue to come down in time, and is going to play an ever increasing role in sort of what diagnostics look like going forward. And there are bound to be new technologies that kind of enter into the picture. But all of these things are focused on pathogen identification. And the assumption there is that if we find the pathogen, we'll know what's causing the infection. Well, there are some challenges with that. There are lots of slides and papers that I can point to. This is an interesting one, I thought. This is the EPIC study, which is now several years old, but it was a study that was designed to try and find the microbiological etiology of pneumonia. They enrolled hundreds of patients, a couple of thousand patients actually, over a few hospitals over a couple of years. And they didn't just do sort of standard of care testing. They did every test that they had at their disposal. PCR for strep pneumo, serological assays, all the tests that they could have at their hands. They didn't do sequencing, which wasn't available at the time. But what they found is that only 38% of adult patients with pneumonia had an identified etiology of their pneumonia. So what does that mean for everybody else? Do they not have an infection? Do we just not find the pathogen? We have no idea. It could be any of those possibilities. And of the ones that had an identified etiology, most of the time it was a viral pathogen. Now, I can tell you clinically, I don't know if there are clinicians in the room, but if you see a patient with respiratory symptoms and you get an X-ray, and there's even, despite the fact that many of them may not have an infection, maybe it's aspiration, maybe it's some other kind of inflammation, or if they do have a pneumonia, it's probably viral in a majority of these cases, but we just don't know. So they all leave with an antibiotic. And this story plays out over and over again. In sepsis, the most severe form of infection, you only find a pathogen in about half the cases. Skin infections, urinary tract infections, a lot of these, you never find a pathogen. And sometimes when you do, you don't know whether it's an actual pathogen or whether it's just a commensal organism, something that could be infecting them or something that could be colonizing that patient. So there are challenges both with poor clinical sensitivity as well as poor clinical specificity, but this has nevertheless been the state of the art. And so there are consequences to that, right? So there's excessive antibiotic use. In the absence of, or in the presence of diagnostic uncertainty, clinicians don't want to worry or they fear about the possibility of leaving an infection untreated and having that patient get worse. And so the default is to give antibiotics. And back in the time when we used to think that antibiotics were harmless, that seemed like a reasonable strategy. You take your antibiotic, and if you get better because of the antibiotic, great. And if you get better in spite of the antibiotic, great. But we know better now. We know that that's not an appropriate strategy, that there are implications for the health of that individual patient, not to mention the public health implications. So lots of inappropriate antibiotic use and lots of consequences to that, both stemming from the inappropriate underuse for patients that actually need the antibiotic, or they're getting the wrong antibiotic, as well as overuse of antibiotic. Again, getting a broader spectrum antibiotic that they don't need or getting an antibiotic at all. All of those things have implications. And so since I started off talking about an infection and what defines an infection, let's look at the other side of the coin and ask whether or not the host can tell us more. And so that's really where a lot of this is going to focus. Now, the reason that there's a lot more emphasis on host response now is because they're enabling technologies. All of these omics, lipidomics, genomics, epigenomics, whatever you want, add an omic to the end of it, and you've got some brand new field of study. And so we've got all of these tools to measure all of this massive amount of biological data, but we also have the analytical tools, the machine learning capability in order to make sense of millions of data points in a very small number of patients. And so that's really opened up a lot of new opportunity within the past, I would say 15, 20 years, that's now evolving to the point of actually getting out into the clinical domain. And so host response, I'll sort of take a step back and think about it from I guess, a historical perspective and how we got to where we are now. Well, the concept of host response has existed for millennia. It's really just how it is that the patient reacts to a particular stimulus. We think about rubor, right? Pain, warmth, swelling, and redness as the hallmarks of inflammation. Now, that could be from anything. It could be from an infection. It could be from any inflammatory process. But those are all host responses. They're not biomarkers per se. They're clinical manifestations. But that's really what a host response is. And as our ability to identify the molecular or the biological basis of those clinical manifestations of host response, as those started to emerge, then we could actually identify something that you could quantify. ESR has been around for over a century, erythrocyte sedimentation rate, just asking how quickly it takes for your blood cells to sort of settle in a test tube. It seems really crude, but it's a marker of inflammation. C-reactive protein, also around for almost a century as an acute phase reactant. Procalcitonin, probably one of the most widely studied but relatively recently identified biomarkers, identified in the early 90s. Almost in an accidental way, where they were really focused on the pro-hormone to calcitonin because the researchers who were working on it were really focused more on calcium homeostasis, and then discovered that in certain situations, particularly in sepsis, it was vastly upregulated and expressed by nearly every tissue in the body. And so individual biomarkers are now sort of entering into the picture. But now, again, you've got sort of the omic revolution in a sense. And here we have the ability to identify a population of patients, take some samples, measure anything and everything, run it through a machine learning algorithm, and then spitting out at the other end is essentially a signature or a biomarker panel of whatever disease or whatever clinical question you're interested in. But of course, you then have to develop assays for, you have to validate. I don't want to oversimplify this. There's a tremendous amount of work involved, but the approach is changing. Rather than going into it in a biased way, in a supervised way, where we say, you know what, I need to understand the underlying biology of this disease. And once I know that biology, I'm going to identify the biomarkers that I think are relevant. And now I'm going to go and target those as potential biomarkers for whatever I'm interested in. Here you go in and you say, I have no idea what's important. No clue whatsoever. And frankly, I don't care about its underlying biology. Because if my machine learning tells me that this combination of biomarkers can distinguish disease A from disease B or C, whatever condition you're interested in, then that's what matters. You can go back and figure out the biology after the fact. But whether it works, that's ultimately the most relevant question. So we're approaching this in a more unbiased, unsupervised manner in terms of the discovery efforts. And so now I'll take you a little bit through some work that we had done applying that sort of omic-based approach in the context of bacterial versus viral discrimination. So this paper gets progressively older every time I give this talk. It's from 2016. It feels a little stale at this point. But we used, the question we were asking is, can we do a better job of differentiating patients who have a bacterial infection from a viral infection so we can use that information to figure out who needs antibiotics and who doesn't? And so we enrolled a couple hundred, almost 300 patients who had suspected infection, as well as a control group who were sick without an infection, and measure gene expression in the form of microarrays. Nowadays, everybody uses sequencing for the most part, but back then, microarrays. And then we developed or used machine learning techniques to identify a signature or a collection of changes in gene expression that allowed us to differentiate the three groups. We developed classifiers, essentially a mathematical equation that took that gene expression information and converted it into some sort of a probability that a patient had a bacterial infection or a viral infection or some non-infectious illness. So for all practical purposes, we had three mathematical equations, three sets of biomarkers to address each of those individual questions, validated using leave-one-out cross-validation, which is probably not relevant, but up there anyway. And then since each patient essentially had three probabilities, the probability of bacterial, the probability of viral, the probability of non-infectious, for simplicity, we just said whichever one is the highest probability, that's going to be the diagnosis. And so we then applied that to the population of patients. And just to sort of walk you through, every patient is sort of one point along the x-axis. So here we had just, about 115 or so patients who we clinically adjudicated as having a viral infection. And in each column, essentially, you have three symbols. A blue circle represents the probability of a viral infection, a red square represents the probability of a bacterial infection, and a black triangle represents the probability, red is non-infectious and black is bacterial. And so one thing to highlight is that these are all patients who had a viral infection, and the blue circles are essentially all clustered up at the top. So by and large, we're able to classify these patients correctly. We got 90% of them correct, 92% specificity, excluding patients who didn't have a viral infection, as some other etiology. And just to sort of highlight one particular case, so this was during the 2009 H1N1 outbreak. This was a camper who was on the Duke campus who basically developed H1N1. And I picked this one out because she had the highest predicted probability of a viral infection, almost 99%, and her probability of having any of the other etiologies was exceptionally low. Well, now we take a look at the patients who had a bacterial infection. Again, clinically adjudicated was sort of our reference method in this case. And here you expect to see the black triangles at the top, and by and large, that's what we see. Now you do see a couple of error, things that could be errors. Here on the far right, you've got a red square up at the top, and the black triangle is down close to the bottom. But by and large, they're falling into the expected pattern. And this is true of gram-positive infections, gram-negative infections, atypical infections. All of these were bacterial. That was the only thing they had in common. And the case I pulled out here, this was a 60-year-old man with a liver transplant, so immunocompromised, who came in with pleuritic chest pain and fever. And he had a CT that basically showed he had pneumonia and ultimately was found to have Staph aureus bacteremia. Sort of lowish probability of having a non-infectious condition. SIRS is a systemic inflammatory response syndrome, essentially just synonymous with a non-infectious illness for these purposes. And then the non-infectious group, this was a very heterogeneous group. Some had like pancreatitis, there was drug overdose, there was dehydration, just a litany of different things. The only thing they had in common is that they didn't have an infection. And so in this case, you can see that, again, the red squares are all clustering up at the top. There's a little bit of spread, but by and large, we're seeing the expected pattern. In the case I pulled out, this was a woman who had non-small cell lung cancer, who came in with shortness of breath, coughing up blood, and low oxygen levels. A CT scan showed an increased size of a mass-like opacity. She was treated for pneumonia as the possibility, but ultimately this proved out to be a progression of her underlying cancer. Was built on a variety of other studies we had done in the context of viral infection and other sorts of infection, and then we went on to validate this in a variety of populations. We looked at sepsis. We looked at then developing platforms that took these signatures and put them on an actual testable platform. So we've done a lot of work over the years, leaning into COVID. There's some metagenomic stuff. There was some microRNA work. All of it really focused on our ability to use host response to help classify and categorize infectious disease. All that's great, but then how do you actually take those discoveries and develop something that you can use at the bedside? And in this context, there are a few different approaches. So here, just to start off, these are companies that we specifically engaged with to take our gene expression signatures and put them on their platforms. Any of you in the clinical micro world will recognize, and EPAASM is actually a local Philly company that we worked with. And none of those collaborations have, for one reason or another, none of them have either gotten to market yet. In some cases, like in BioFire's case, they decided it was not a priority, and so it got shelved, very sadly. But the tests worked on all of these systems. There are other companies that our work was not necessarily specifically involved with, but that are in this space. Informatics is a spin-out from Stanford. And then Cepheid, where I'm currently affiliated with, has some gene expression-based tests that they're currently developing, including in bacterial versus viral discrimination. And then there are protein-based signatures. So actually, these two are both FDA cleared and on the market. FebriDx is a lateral flow test based on of C-reactive protein and MXA, myxovirus A protein, which is upregulated in viral infection. And then MEMED measures three proteins, CRP, TRAIL, and IP10, that in combination give you a sense as to whether or not the patient is having a bacterial infection or something else. So there are tools that are already out there and then others that are in development. The point that I want to make here is that we are starting to actually make some headway in terms of our ability to get from this discovery phase to something that's actually usable at the bedside, which is pretty exciting. So now I'm going to pivot a little bit and tell you about some of the work that's now happening at Danaher in the host response space. And so one of them is in the context of TB, and specifically latent TB. So there's a very large spectrum of TB, but I'm going to sort of narrow in on kind of 3 states. One of them is the non-infected state. And in that scenario, there are a variety of tests that one in theory could do to put a little fluid under your skin, and you come back 2 days later to have it read. There are blood-based tests looking at the release of interferon gamma or IGRA. Of course, culture, sputum smear, PCR, and then based on the combinations of those different tests will indicate whether or not you have no infection, a latent infection, which is about 90% of the population who's been infected with TB, where you actually have viable TB in your body, but it's not causing any clinical disease. And those people are at risk for reactivation at some point in their lives, which is why there's a big push to try to identify those people, because if you can treat them for their latent infection and they never convert to active TB, then they can never spread their TB. In the developing world, most of the focus is on active TB, mostly because of resources, because so many active TB that's really where the emphasis lies. And for any of you who are involved in healthcare, you probably get a latent TB test on an annual basis. So it's different markets, different clinical needs, but nevertheless, currently, the emphasis is on sort of jump ahead here. In terms of latent TB, again, historically, it's been these skin tests, but now there are these interferon gamma release assays. And generally, the way they work is under the assumption that if you've been infected with TB, even if it's latent, you've developed an immune response to it. So if we take some of your blood that includes your T cells, and we incubate it in a tube that's got TB antigens, it should react. And that reaction will result in the release of interferon gamma. So blood is collected into tubes that have TB antigen, it's incubated generally overnight, and then the amount of interferon gamma is measured the next day. And then depending on that amount, will determine whether or not you do have an immune response or not. And if it's positive, then you've got latent TB. And in the absence of clinical symptoms, you get treated for latent TB. And there's a big transition from skin tests, even though they're cheaper, they're a little less reliable, and they do require the patient to come back and get it reread, more to a lab-based methodology. And so what we're asking at Cepheid is whether or not we can approach this from a gene expression perspective and whether or not that might allow us to do a little bit better. And so we set out to do that. I initially had, and this is actually all before I joined the company, so I can't take any credit for this, but they started off identifying a panel of about 26 different genes that were differentially expressed in latent TB and were hypothesized to potentially be of relevance. Now, I'll mention that the Cepheid cartridge can measure up to about 10 mRNAs in a quantitative way. So we had a cap on how much we could potentially measure. Based on some in vitro testing, we narrowed that down to about 15, and ultimately, after some testing in some subjects, narrowed that down to 8 that eventually made it onto a Cepheid cartridge. And so the workflow is you collect blood in just one tube, you stimulate it with TB antigen, you load it onto a gene expert cartridge, you stick it in the system, and you get results in under an hour. And this is just an example. This is a pilot study just presented at TB Union a couple of months ago. And this is an example of IL-2. And here we define active TB as having a positive culture or positive PCR. Latent TB is defined by another one of these IGRAs, just developed by, in this case, QIAGEN. And then there were some patients who had no TB. And what you can see in the scatter plots is generally that the expression of IL-2 is, in this case, these are CT counts. So a lower value means a higher level of expression. So you see lower levels of IL-2 mRNA in active and latent TB relative to the non-infected group. The same thing with the interferon gamma mRNA. When you look at these in terms of a histogram, it looks like the latent and the active overlap. The non-infectious group is separate. The same is true for the interferon gamma. But there are other genes on this panel that do a better job of discriminating latent and active. And we're not just focusing on one single biomarker, and this is the advantage of having a signature. is that whereas one biomarker may not be enough, when you have essentially an algorithm that integrates the information across multiple biomarkers, now you actually have the ability to differentiate it with just a single biomarker approach. And so when we model this, we actually get near perfect separation of the active TB group from people who didn't have active TB, latent TB from people who didn't have latent TB, and the non-infected group. And currently, there's no test available that can differentiate active from latent TB. It's basically a clinical diagnosis. So not only can we identify latent TB, which is what the test currently can do, but we now add that additional capability of differentiating active from latent. So I thought this was pretty cool, just in terms of that added capability. This is still very preliminary, and no doubt that once this evolves further, it's not going to be quite this perfect. But it is, I think, nevertheless exciting in terms of the opportunity. Next, I'm going to pivot to sepsis and some more work we're doing here. So I referred to sepsis earlier. This is a sort of a severe, life-threatening infection caused in large degree by the body's response to the infection. You can take a whole bunch of people who have an E. coli urinary tract infection. Some of them are asymptomatic, some of them have mild symptoms, and some of them are in the ICU on pressors. It's the same bug, the same site of infection. The only thing that differentiates one person from the other is their response to the infection. So sepsis is really driven by that patient's immune response to the infection. Obviously, the pathogen plays a role. Some pathogens are more predisposing to causing sepsis than others are, but it's the immune response that really drives this. And there are all sorts of really terrible numbers here. And the point is that, sepsis is a bad condition. It's highly prevalent. It kills lots of people. And there's not a ton that we can do about it, unfortunately. There have been greater than 100 sepsis clinical trials evaluating a variety of therapeutics. And I've highlighted in red all of the ones that work. So nothing has ever been shown to be successful in treating sepsis. It's low, we'll give you medicine to raise your blood pressure. Your oxygen's low, we'll give you oxygen. You have an infection, we'll give you an antibiotic. But there is nothing that works that actually treats the underlying immune response. And the root of the problem is the fact that sepsis is, you can almost think of it like cancer. If we were to say that cancer is one disease, that might have worked 100 years ago. But we know that cancer is not one disease. Cancer is hundreds of diseases. Even within one particular tumor, there's probably different biologies represented. And sepsis is the same thing. It's not one disease. It's a syndrome that manifests in this form of critical illness. But there are lots of different biological abnormalities that are going on that ultimately manifest as sepsis. And so those are going to be driven by pathogen factors, host factors, environmental factors, the microbiome, a variety of different things. And so the idea that you can pick one drug and expect it to treat all of those underlying biological abnormalities, it makes sense that all of these trials have failed. And so the thinking now is that what we need to do, and this is just sort of describing that in the same way, I'll just sort of skip over this. The thinking is what we need to do is we need to define those underlying biological subtypes and focus on treatments that treat that particular group's biology. And maybe you find a subtype that exists, it's got a prevalence of maybe 20%, but you've got a drug that can work wonders for that 20% of the sepsis population. And maybe for the rest of the population, it doesn't do anything, or maybe it even harms them. You'll end up with essentially a negative trial. But if you find the group of people who are going to benefit, that's now the thinking in terms of how we can actually finally start to make a difference in the management of sepsis, specifically with respect to the underlying immune response. And so the idea is that we can use a variety of different factors, multimodal inputs, transcriptomics, proteins, whatever the case is, clinical variables to identify treatable traits. And Danaher has a variety of technologies that we're developing in addition to the existing capabilities in the forms of high sensitivity immunoassays, advanced imaging-based hematology, molecular diagnostics, clinical decision support, other ways of trying to enable that approach. And so in order to achieve that, we embarked on a collaboration with Oxford University. There's a group there led by Julian Knight, who's been focusing on identifying these subtypes in sepsis using a variety of approaches. But one in particular uses gene expression. And this is now, again, a little bit of an aging study, 2016. But this was really the paper that kind of set off a lot of research that subsequently followed. And what they did is they identified 265 patients admitted to the ICU with pneumonia. They've also subsequently looked at other types of sepsis, but these were all patients with sepsis. They did gene expression analysis. And again, they said, well, we don't know what are the relevant genes. We don't know the relevant pathways. Let's just see what the data tells us. And so they started off by doing a clustering analysis. And that's that figure all the way on the bottom left. They essentially just asked, how many different groups can we define based on gene expression patterns? And depending on where you cut things off, in terms of where you draw that horizontal, you can have any one of a number of groups. If you make the cutoff all the way at the bottom, then every patient has their own grouping. But at a high level, they can see that there are two major groups. Group on the left, sort of outlined in that blue color, and the group on the right in the red, which is sort of the majority of the patients. They can visualize that with a PCA and that sort of second plot there, and they can see that the red and the blue groups separate from each other. And then what they did is they said, well, does that have any meaning? Like, what do we care if there's multiple groups? What's the significance of this? And they called these sepsis response signatures, SRS. And they found that patients who had SRS1 were sicker, they had a much higher mortality rate, higher incidence of organ failure, they were in the ICU longer. Every bad thing you can possibly think of, they were more likely to have it. Now, this wasn't a guarantee. It's not like if you had SRS1, you were going to die. Mortality was, you were still, I guess just looking at the curves there, this is now looking at mortality or survival in plot E, and then F is essentially just a validation cohort. But the SRS2 group is shown at the top, much higher survival, and the blue group is the patients who were assigned to that SRS1 category, much higher mortality rates. And so then they did lots of different studies. I'm not showing you all that data, but one of the things that I am showing you here, which I think is pretty cool, it speaks to the actionability of that information. All of these tests, you measure all these different biomarkers. It's all well and good. Somebody can give me a test that says this is a patient that has a higher likelihood of having a bad outcome. But what am I going to do with that information? If this patient has sepsis, they're in the ICU, I'm still going to treat them aggressively. So where does the actionability come from? So what they did is they looked to a study that had already been completed called the VANISH study that looked at different pressors and also the administration of steroids. So it's sort of a 2x2 design. And they went back to samples that were banked from that study, and they measured these SRS gene expression signatures in this population. And what they found is that if you were in the SRS1 group, this is already the group that's worse off, if you gave them steroids or placebo, it made no difference. But if you were in the SRS2 group and you got steroids, your mortality went up significantly. So they demonstrated a treatment interaction effect. And based on the underlying biology, there's reason to believe that there are likely to be treatment interaction effects with a variety of other agents. And as we saw with COVID, there are a lot of immunomodulatory drugs that entered into the market, IL-6 inhibitors, IL-1 inhibitors, and these create opportunities to now potentially target some of that underlying biology in patients who have the underlying defects.
Just a question. This study, this transcriptomic signature study, is all based on image.
Yes.
Did they try to compare it to viral, bacterial, and non?
It's a good question. So their goal was not to differentiate bacterial from viral from non-infectious. But what they did do is they validated in patients with viral sepsis, with COVID, and those same groups exist and the same patterns exist. And so whether or not your sepsis is due to a bacteria or a virus or a fungus, it doesn't matter. This is focusing on the host's immune response to the sepsis.
I understand that the S2 contains all of the different ones. That's what I'm trying to do.
No, in this case, these were all bacterial infections. Yes, in this case, they were all bacterial, but they've also seen the same patterns in patients with viral sepsis, or whether you're viral, but it doesn't differentiate between whether it's bacterial or viral. And while I don't have I'm not showing you the data here, the same sort of concept has been applied to ARDS, acute respiratory distress syndrome, which can be infectious in its underlying cause or trauma or burn or aspiration. Lots of things can result in ARDS. And as it turns out, there's two major groups in patients with ARDS. They're referred to as hyperinflammatory and hypoinflammatory. And there are both gene expression as well as protein biomarkers that can differentiate those groups. And they've seen treatment interaction effects with steroids, with statins, with different fluid strategies, with different ventilatory strategies. So there's clear evidence that we're starting to be able to subtype these complex critical illness syndromes in a way that can ultimately be sort of lumped into treatable or identified as treatable traits. And there also seems to be a lot of overlap between sepsis and ARDS, for example. So this isn't necessarily a unique strategy for sepsis, but could be more broadly applicable. And so what we're doing as part of this collaboration is trying to develop a test that measures this gene expression signature to be able to stratify patients as either belonging to this SRS1 or SRS2 group. Because once we then develop the tool, then we can start to apply it more broadly, right? Right now, all of this is research-based. They collect a sample, they bank it, extract RNA, run sequencing, not clinically applicable, and just based on resources, there's only so much you can do with that. But if we can develop a test that they can run every day on whatever patient they want, essentially as close to the patient as they want to get, now they can really start to explore what are the ways in which a test like this could be useful. What are the drugs and the treatments that we might be able to link to this new phenotype information that we're providing? And we're doing this through something called the Danaher Beacons. So this is a relatively new program at Danaher, really just in the past year, as something that we've developed to identify essentially opportunities for sponsored research. So Danaher identifies areas of prioritization, but where we lack the capabilities, we lack the insight, we lack the resources, and it gives us a mechanism to reach out to people by and large in academia who are doing some earlier stage innovation where there's a lot of conviction and belief and confidence that this will eventually result in something that can be developed in sponsored research that can ultimately sort of feed back and support obviously the clinical community, but somewhere where Danaher can get involved in an earlier stage. And so we entered into a beacon with Julian Knight. And again, the goal is to develop a point of need test that enables the translation of the science that he's developed into direct patient benefit. It allows us to move away from a clinical syndrome of sepsis to defined subtypes or endotypes and ultimately treatable traits, using that information to guide therapeutic interventions based on the underlying immune response that we can then use to guide care pathways, patient management. There's other data I'm not showing you that shows that this is gene expression signature can also identify patients who are developing sepsis before they've manifested with clinical signs of sepsis. This is really in an at-risk population, like post-op patients, and enable the development of biomarker-led clinical trials so that we can really start to get to the point where we can now make a difference or identify something that can treat patients with sepsis. And so, again, many of you are familiar with Cepheid, but for those who aren't, I'll just tell you a little bit about Cepheid's technology. So Cepheid is a molecular diagnostics company, and they got their start. I'll sort of go through the sequence. The little thingy on the left is essentially a Cepheid cartridge. Everything is contained within that cartridge. So the sample goes in, and all the magic happens in there. The system itself is highly modifiable in the sense that there are modules. Each of these little slots where one of these cartridges goes in is one module, and they can exist anywhere from a single module design all the way up to an 80 module design. And so a lot of flexibility in terms of what it can do, but each is essentially a separate and independent reaction. Following COVID, Cepheid is now the largest molecular diagnostics company in the world, greater than 50,000 systems worldwide. In the US, more than 20 IVD clear tests. So lots of statistics about how big Cepheid is. They got their start, interestingly, 20 years ago, developing an assay for anthrax that they put in post offices. So those of you who are old enough to remember, there was an anthrax scare around that time, and white powder showing up in envelopes, and then lots of sort of copycats thereafter. And so they wanted a test that could essentially rapidly identify the presence of anthrax in a sample. And so Cepheid provided that. After that, their first clinical test was a Group B strep test. An MRSA test emerged shortly thereafter. And I know being at the VA had a national mandate to screen every patient for MRSA before they got a bed assignment in the hospital. And you couldn't do that with agar. So Cepheid essentially got introduced to the VA system specifically for MRSA testing. But then, there's lots of different assays in here as well. There are oncology applications as well. This is just sort of a highlight of some of those. I think the most recent one to get FDA clearance was a multiplexed vaginal panel. to identify the etiology of vaginitis. Some of the characteristics of Cepheid that enables the use of that technology for mRNA expression analysis is that it's multiplexed. So like I had highlighted, we're not just measuring one mRNA in these signatures, but there's a signature, there's a panel of mRNAs that we're measuring. And depending on the complexity of the clinical question, sometimes you can get away. There are some things that this system is not suited for, but for things that we can identify with a smaller number or limited number of mRNAs, this works. It's quantitative. It's based on CT count, and so we can use that PCR cycle threshold in order to get a sense as to how much of each mRNA is present. Because with these host response signatures, we're not trying to figure out if the mRNA is present or absent. This is different than a pathogen assay. We know that the mRNA is expressed in every human sample we're going to get. The question is in what amount relative to a control population. So we need it to be quantitative. And there are already gene expression-based assays that Cepheid's developed for oncology applications. And then ultimately, all of that data has to feed into an algorithm that's sort of part of the system, part of the test, in order to spit out an interpretable answer. And so I'd already told you about the bacterial versus viral work that Cepheid is doing, or I alluded to it, I didn't show you data. There's the TB work, and now there's sepsis work. And so we're leveraging that capability in order to support this collaboration to develop these assays. And so now I'm going to pivot. I'm actually nearly done. I'm going to pivot a little bit to talk about some work that's happening at Beckman Coulter. Beckman Coulter is a large company in and of itself that has areas of emphasis in hematology, urinalysis, clinical chemistry, immunoassay, microbiology, and now they've recently added a new business unit that's focused entirely on clinical decision support. And the idea is that as we develop ever increasingly complex assays and tests, and an ever increasing number of tests, and you couple that with continuous vital sign monitoring and all sorts of other inputs, it becomes increasingly difficult for the clinician to know what to do with all of that information. And so clinical decision support, whether it's driven by AI in some cases, or just sort of more standard machine learning clinical practice, to be able to synthesize all that information. And so essentially Beckman is building, for all practical purposes, a platform, an architecture that is fully integrated with all of the different complexities of medical record systems and the LIMS, the labs, in terms of delivering, aggregating, synthesizing, and delivering interpretable and actionable information to the clinician. And we see this as ultimately being a really important part of how we are ultimately going to develop and deliver complex assays like a host response assay that we want to link to some kind of action for the clinician to take. So we don't have it there. There's one commercial product that's out there. It's actually an ED triage system. If you're curious, I can talk about it later. But it greatly facilitates how patients are triaged when they show up in the ER to be able to get the right kind of care. And it decreases time in the ER, increases throughput, decreases. Developed at Hopkins, spun out of Hopkins as a company that was then acquired by Beckman Coulter. So with that framework in hand, there's a lot of things that we're building on top of that. A lot of them focused on sepsis, focused on acute kidney injury, on cardiovascular events, on dementia. So there's a lot of applications and ways in which we can use this architecture to ultimately expand on and facilitate care. So I think that's all I've got, and stop there and be happy to take whatever questions.
Great point. So go back to the other slides and the microarray, other different things back here. So how well has have to perform for clinicians to actually use it? Because test that has, say, let's just say it's 95% sensitive and 97% specific. It sounds like a good number, but that may not be good enough for someone to say, I'm not going to treat this person with antibiotics. So how do you attack that?
Yeah, so that's a question we think about a lot, and it's extraordinarily dependent on the context. So for example, if you just look at outpatients who are presenting with respiratory symptoms, 80% of them are viral. So if I had a completely worthless test I'd have a sensitivity of 80%, right? Which is not that bad. So a lot of it is going to be dependent on your pretest probability. So there's that. But then there's also the clinical variables. What are the risks of me getting it wrong? So again, if you're in an outpatient environment and you incorrectly say that this patient doesn't have a bacterial infection and you withhold antibiotics, then chances are they'll be back in another day or two and say, you know what, I'm not getting better. Or you know what, now I have a fever. You say, okay, here's your antibiotic. And then that's okay. Yes, there's always a risk that that patient may progress from mild illness to all of a sudden septic shock in 2 days. But that's so exceedingly rare that in an outpatient environment, a test that's got sensitivities and specificities in the 90s, that's fantastic. Like that could really be impactful. Ultimately, you need the clinical utility studies to prove that because clinicians are always going to hang on those few percentage points and they're going to think about the anecdote of, well, what if I get this wrong? But it also has to be interpreted in the context of what they're currently doing. How many patients do you actually not know how to diagnose correctly? You think you know. It's one of these scenarios where everybody else gets it wrong, but I know I'm doing it right. So it's all those other idiots out there that are overprescribing antibiotics, but I know when to give them. And so, the other way to think about it is not in terms of sensitivity and specificity, but in terms of incremental diagnostic value. How much more is this adding relative to what I'm currently doing? Now you put that in the ICU where somebody's got a blood pressure that's in a toilet and you run a test that says, there's no infection present. Nobody in their right mind is going to withhold antibiotics, nor should they. So it's very much context dependent. And all of these tests are also being sold as aids in the diagnosis of, to be used in conjunction with, right? So they're de-risking a lot of that because none of them are going to be able to stand on their own as the absolute diagnostic test for some condition, but it's one piece in an array of other things that we use. Or like, take a white count. You think a white count could get through the FDA with an AUC of like 0.55 for infection, but they measure it every single day in the hospital, sometimes multiple times a day. And does that have value? I don't know. I mean, I think the kinetics of a white count is more useful than an absolute white count. But I see trainees who measure a white count and it comes back at 12 and they're like, what do I do with that? It's like, I don't even know why you ordered it, right? Are you going to treat this patient with antibiotics? Yes, I think they have an infection. Okay, well then what difference does it make? If their white count is normal or it's abnormal, you're still going to treat them with antibiotics. So it also gets to the actionability of the information that you provide. That was a very long-winded answer, but in short, it depends.
It's tough not to crack.
It is.
When looking sort of being the initial investigation and development of these panels, are you typically using sort of a normal non-immunocompromised host versus who you think is sick? And how do you get towards developing diagnostics for immunocompromised and immunosuppressed patients who may have more severe infection, but not the same one?
We generally focused on, initially, we just sort of took all comers. I think if they were like neutropenic, we excluded them. But we've also done some studies where we focus on a sort of a non-immunocompromised population. There are advantages and disadvantages of both approaches. If you focus on a non-immunocompromised population, you'll probably get a cleaner result, might perform a little bit better, but ultimately isn't as applicable to the people who might need it the most. And you're also selecting for biomarkers that may not be relevant. There may be different biomarkers that are relevant in an immunocompromised population. So there's some advantages to keeping it dirty from the beginning and having a more heterogeneous population. Because one general thing that's true when you use these machine learning methods to do biomarker discovery is that you should never try to apply them in patients who were not represented in the discovery process. So it's important to think about who you ultimately want to use this test in when you're doing that initial discovery work. In our case, we subsequently validated it specifically in an immunocompromised population, the bacterial versus viral component. It worked, not quite as accurate as in a non-immunocompromised population, but still gave valuable information.
So, can I pick back on that question? So
Great question, Courtney. How are you approaching that like regulatory advice? So, the failure or the challenge of some, take the IGRA that you showed, right? The original IGRA did not perform well with people with advanced HIV. And now there's a new version of the IGRA better. Do you think that the algorithm and the multiple target approach will overcome that? Or do you think that you see us having restricted populations in which we can use that? Or do you see like blanket approval with like additive carve-outs where we recognize they don't work as well? And I ask because that once it gets released into the wild, people use it like however they want. And there is no like cohort in which this test is allowed to be ordered unless you build really strong EMR rules.
Yeah. It's too early in that project for me to know whether or not there are certain populations that it doesn't work as well in. But the HIV population is obviously critically important to analyze, particularly if you see this being used outside of the US, where TB is endemic and obviously HIV is as well. Very often, and now I'm going to sort of give you a generalization. Very often when a test is being developed, the initial population is going to be one that's cleaner. Because if you include a population where you're going to get messier results, then you might fail to meet your pre-specified criteria, and ultimately your test doesn't get approved at all. And so generally speaking, the focus is on patients who are less likely to have issues in terms of their immune response as far as what the test is measuring, unless the FDA has specifically said, you need to provide data on how this performs in an HIV positive population, in which case, yes, you've got to do that. And then what often follows are claim extensions. They say, okay, well, we've shown it works in this population. Now we want to see how it works in this other population. And so we're going to do a separate study and submit that to get a claim extension to now say that, yes, it works in this group and in this group, but it may end up being piecemeal. I think it very much is test dependent and situation dependent.
Great talk. So it's funny that you mentioned the protein-based disease because when I think about TB, one of the first features is neuron response. So I'm just trying to see how you envision the AE versus active phenotype. Again, if you're going to the endemic regions, a lot of people will be latently infected, and if they're active, you can get it through this phenotype. Right? So where's the application there in combination with their IGRA?
So the ideal would be that it wouldn't be in combination with an IGRA test, but it would replace an IGRA test because it can function as an IGRA test. So and a lot of it is dictated by the clinical scenario. So if a patient comes in and they don't have any clinical disease and you have no suspicion for active TB, then for all practical purposes, it's a latent TB test. You run it, and if it comes back positive, then you assume they have latent TB. But the scenario where I think of this in anecdotes. So when I was an ID fellow, I don't know how many times I would get called with a team that had admitted a patient who looked like they had a pneumonia. Maybe it was cavitary, maybe it was cancer. They didn't know, but you see something in the lung and it's cavitary. Is it TB? And so then they plant a PPD or they run an IGRA and it comes back positive. And they're like, what do we do? Like, well, when you're done treating the patient, send them to the health department so they can get treated for their latent TB. Because they never actually came back as actual TB. They just had latent TB and some other clinical syndrome that was manifesting. And so it particularly in areas where TB is more endemic, the ability to differentiate between TB as the source of their disease versus just a bacterial pneumonia or something else entirely becomes a clinically useful tool. So it's not to say that there is a massive market of people that need to be differentiated from active from latent, because that's generally a clinical decision, right? If they're sick or not. Is one of the main differentiators. But there are scenarios where that capability becomes very valuable. Now, if you can get a sputum, yeah, you use your conventional microbiological tools, and that'll tell you whether it's active TB. But what if it's miliary TB or extrapulmonary TB or it's pediatrics and you can't really get a sputum sample? So there are scenarios where having a host response or a non-microbiology based diagnostic can also be very valuable.
There was an old Lancet paper. They look at blood signature for TB. And it was longitudinal, so they were able to predict reactivation of TB up to 6 months. Mm-hmm. So that I think that would be helpful. That you can start treatment.
Yep. And Cepheid develop, I think, licensed that signature and developed that as a separate. I didn't talk about it here. And there are actually a number of papers that are evaluating, they call it the TB finger stick. And a lot of that work that was published was looking at kind of publicly available data or sequencing, microarray, whatever they used. When you actually develop a test, it doesn't, and try to validate it, it doesn't validate quite as well as it did in the discovery, which is often the case. But yes, and there are some papers that are looking at that TB host response in different populations in young children and adolescents and adults. It's not a perfect test, but again, there's additive diagnostic value, particularly in those cases where you can't get sputum, for example, and you're stuck without a diagnosis.
We're going on with that. So, with the genetic variability of human populations, with their different environmental exposure and maybe chronic inflammatory low-grade inflammation, how big a problem is that to distinguish either between different diseases or different time point of their progression?
Yeah, so this relates to the question about the immunocompromised, which is that I'll give you an example. There's a company called Immune Express that developed a sepsis versus SIRS gene expression signature, and they developed that signature in the Netherlands and in Utah. It was the patients that they drew from. Now you can all kind of gather what patients from the Netherlands and Utah look like, and so then they developed this signature, and they developed a test, and then when they submitted their FDA filing, 80% of African Americans would be sepsis. 40% of healthy Asian Americans would be diagnosed with sepsis because they developed their signature in a patient population that was never represented in the discovery. So to your point, if you've got people from all over the world that you're thinking about applying tests to, you need to make sure that you have that ethnic, that genetic, that geographic, that microbiological heterogeneity represented in your population so that when you pull out the genes or the signature, the biomarkers that represent your disease of interest, all of those other factors fade into the background, and you're only focusing on the things that are common to all of the people that are represented in your population.
I think the idea of personalized medicine, maybe it'd be good to have that everyone has baseline level. Then when you get sick and you know what to essentially change.
That would be great.
That would be the idea to, instead of weighing yourself, maybe,
Yeah, so a future state where NGS is cheap and fast, right? You can like Gattaca, if anybody watched that movie.
I want to ask one, I know it's getting late.
You've had your hand up. Go for it.
Yes, sorry about that. I just think there's so much power in this analysis, and in my lab, we work on Clostridium difficile, and in particular, we're trying to develop vaccines for prevention of disease and maybe even prevention of infection. And so as I think about and listen to the presentation today, I think about it would be so useful for C. diff where we have asymptomatic carriers versus those that go on to have symptoms versus those that go on to recur.
Yeah.
And there's an aging underlying piece to it. But in our lab, we have a cohort from Hahnemann Hospital where we've been following patients before hospital closure. But we see this inflammatory signature that's actually seen in asymptomatic patients versus the ones that actually going to get sick. And I'm thinking, have you considered looking at C. diff populations to use this as a way to characterize and have predictive tool for those that would go on to it?
We tried, but we just didn't have enough C. diff patients to make any sense of what we got. It was a dozen or so, but that was like a little. But we tried to ask that question, whether or not we're seeing the same bacterial host response in patients with C. diff, since it's a toxin-mediated disease, clinical disease. And so are they going to have the same or a different host response? So I don't know.
Powerful, because we would predict those that would get sick, recurrent, and then it would even help us with vaccines with correlates and immune.
Yeah. A lot of opportunity. Thank you. Yeah.
Here with that, I think we're going to post one more time. Thank you so much.
Yeah.